Welcome to this introductory course in cell biology. This video is part 2 on proteins. Today, we will discuss protein folding. The folding of a polypeptide can be determined by many different sets of weak non-covalent bonds that form both between polypeptide backbones and between amino acid side chains. There are 4 types of these forces. One is hydrogen bonds, which can form between a hydrogen atom and an electronegative atom. It is relatively weak in the aqueous environment. Next, electrostatic attractions. Electrostatic attractions can occur between fully charged groups or between the partially charged groups on polar molecules. Charged groups are shielded by their interactions with water molecules, so electrostatic attractions are quite weak in water. Ions in the solution can also diminish the strength of this force. Van der Waals attractions. Van der Waals attractions occurs when 2 atoms are situated within a close distance. The 2 atoms will be attracted because of the transient shift in electron density. And finally, the hydrophobic clustering force. Hydrophobic groups, including the nonpolar side chains of amino acids, will cluster in aqueous solutions because water molecules repel these hydrophobic groups to minimize their disruptive effect on the hydrogen-bonded network of water molecules. These forces may be weak, but their combined strength can hold protein tightly in shape. An important factor governing the folding of any protein is the distribution of its polar and nonpolar amino acids. Nonpolar side chains tend to cluster in the interior of a protein because of the aqueous environment inside the cell. In contrast, polar groups tend to be distributed outside of a protein so that they can form hydrogen bonds with water and other polar molecules. But in some cases, polar amino acids are buried within the protein, and they are usually hydrogen bonded to other polar amino acids or the polypeptide backbone. Proteins fold into a conformation of the lowest energy, and most proteins fold up into a single stable conformation. However, this conformation changes slightly when the protein interacts with other molecules. Although proteins can fold themselves into a correct conformation without assistance, chaperones often take part in protein folding. In the crowded conditions of the cytoplasm, chaperones are required to prevent the temporarily exposed hydrophobic regions in newly synthesized protein chains from associating with each other to form protein aggregates. For protein aggregates, we will discuss in the next video. Two regular folding patterns are often found within the proteins. One is the alpha helix, and another is a beta sheet. These 2 patterns are particularly common because they result from hydrogen bonding between the NH and CO groups in the polypeptide backbone without involving the side chains of the amino acids. Thus, although they are incompatible with some amino acid side chains, many different amino acid sequences can form them. In alpha helix, the NH group of an amino acid forms a hydrogen bond with the CO group of the neighboring amino acid, which is located 4 peptide bonds away in the same chain. Like in this diagram, all the NH groups point towards the N terminus, and all the CO groups point towards the C terminus. This gives polarity to the helix, with the C terminus having a partial negative charge and the N terminus having a partial positive charge. The hydrophilic polypeptide backbone is hydrogen bonded to itself in the alpha helix and is shielded from the hydrophobic lipid environment of the membrane by its protruding nonpolar side chains. Regions of alpha helix are abundant in proteins located in cell membranes, such as transport proteins and receptors. Portions of a transmembrane protein that cross the lipid bilayer usually cross as alpha helices composed largely of amino acids with nonpolar side chains. When two or more alpha helices have their nonpolar side chains on one side, they can twist around each other with these side chains facing inward, forming a structure known as coiled coil. Proteins that form coiled coils typically have nonpolar amino acids at positions A and D. These long rod-like coiled coils provide the structural framework for many elongated proteins like keratin. Beta sheets consist of beta strands connected laterally by hydrogen bonds, forming a pleated sheet. Amino acid side chains in each strand project above and below the plane of the sheet. Beta sheets can form from beta strands that are parallel, or from the antiparallel strands. The cores of many proteins contain extensive regions of beta sheet. Four levels of organization in the structure of a protein. The amino acid sequence is known as the primary structure. Stretches of the polypeptide chain that form alpha helices and beta sheets are known as secondary structures. The full three-dimensional organization of a polypeptide chain is referred to as the tertiary structure. If a particular protein molecule is formed as a complex of more than one polypeptide chain, the complete structure is called the quaternary structure. There is an organization distinct from these four, the protein domain, which is a substructure produced by a part of a polypeptide chain that can fold independently of the rest of the protein into a compact, stable structure. It is the modular unit from which many larger proteins are constructed. Proteins can be classified into many families. Once a protein had evolved and folded up into a stable conformation with useful properties, it diverges and can be grouped into the same family because after repeating rounds of gene duplication and diversion, proteins with the same origin can be traced back. In general, the structure of the different members of a protein family has been more highly conserved than the amino acid sequence. In many cases, the amino acid sequences have diverged so far that we cannot be certain of a family relationship between two proteins without determining their three-dimensional structures. Most proteins are composed of a series of protein domains. Such multi-domain proteins are believed to have originated from the accidental joining of the DNA sequences that encode each domain, creating a new gene. In an evolutionary process called domain shuffling, many large proteins have evolved through the joining of pre-existing domains in new combinations. The protein domains has been especially mobile during evolution. These seem to have particularly versatile structures and are sometimes referred to as protein modules. Each of the domains shown has a stable core structure formed from strands of beta sheets from which less ordered loops of polypeptide chain protrude. The loops are ideally situated to form binding sites for other molecules. Such beta sheet-based domains may have achieved their evolutionary success because they provide a convenient framework for the generation of new binding sites for ligands, requiring only small changes to their protruding loops. The three domains have their N and C terminal ends at opposite poles of the domain. When the DNA encoding such a domain undergoes tandem duplication, the duplicated domains with this inline arrangement can be readily linked in series to form extended structures. Such structures are especially common in extracellular matrix molecules and the extracellular portions of cell surface receptor proteins. Others, such as the Kringle and the SH2 domain, are of a plug-in type, with their N and C termini close together. Such domains are usually accommodated as an insertion into a loop region of a second protein. Pairs of domains are found together in many proteins. More than 2/3 of all proteins consist of 2 or more domains, and the same pairs of domains occur repeatedly in the same relative arrangement in a protein. And these two domain combinations arose late in evolution through domain shuffling. Okay, in the next video, we will discuss the shape and structure of proteins.